In this lesson of Chapter 4, we're going to be talking about the nucleus and also the organelles that are involved in the endomembrane system of a eukaryotic cell. Recall that only a eukaryotic cell would have a nucleus. This is the location where the majority of that cell's genetic instructions are contained and further carried out by the ribosomes. Let's take a look at the nucleus and its associated structures. As shown here, the nucleus has several different aspects to it. The entire structure here, shown as the purple, would be the nucleus. Adjacent to the nucleus is the rough endoplasmic reticulum. And notice how it has the little red dots. Those are ribosomes. So we'll come back to that. But starting with the nucleus, the innermost area that's shown as like a dark purple, that is the nucleolus. And sometimes when you look at a cell under the microscope that's been stained for to adhere to the DNA, you might see that dark spot, but not always. So the nucleolus is the site of ribosomal RNA synthesis. Also inside the nucleus, you see these kind of like dark strands, kind of looks like veins within the nucleus. That's the chromatin. Chromatin, if you take a closer look, is shown here, the bottom right. So the blue strand represents the DNA, and that DNA is tightly coiled around proteins called histones. And the purpose of these histones is to help consolidate the DNA so it can pack into that nucleus. Now, at times during the cell division process, that DNA will uncoil from those proteins, unravel as chromatin, and turn into, it'll still be DNA, but it will appear differently. And we'll look at that when we study the processes of mitosis and meiosis in a later chapter. So chromatin is this association of the DNA strand wrapped around those histone proteins. Okay, so when in the nucleus, that's the state that the DNA will take, chromatin. Okay, on the outer surface of the nucleus is the nuclear envelope. It is a double membrane. It has an inner and an outer membrane. And there are also pores all over the surface of the nucleus. And these pores are going to allow messenger RNA to leave the nucleus, holding on itself parts of the genetic code that are going to actually encode for proteins. Now, those proteins are going to be synthesized either on the ribosomes of the rough ER or in ribosomes that are freely floating in the cytosol. The next slides emphasize in writing what I've just mentioned. So I'm going to pass over those. However, if you want to pause and take these notes, feel free to do so. Okay, so let's continue the story of what would happen to something leaving the nucleus and going into the adjacent rough endoplasmic reticulum. So that something would be a messenger RNA, carrying with it a piece of the genetic code in a form that can be made into a polypeptide, and ultimately that could become a protein. Now, there are other things that are also made by the cell using the genetic code besides proteins. And so as we now talk about the endomembrane system, we're going to see what are some of those substances that are specifically for this system packaged up to leave the cell. So this is an overview of the endomembrane system. It includes the nuclear envelope, the endoplasmic reticulum, both rough and smooth, Golgi apparatus, lysosomes, vacuoles, and the plasma membranes. And these components are either continuous, connected with membranes, or through transfer by vesicles, which are surrounded by membranes. And hence, this is why these series of organelles are called the endomembrane system. So one of the first organelles involved in the endomembrane system after the message leaves the nucleus, and so the nuclear, let me step back actually to just mention again that it's the first actual, I don't want to say organelle, but the part of the organelle involved in the endomembrane system would be the nuclear envelope. The membrane, you see how it connects, the purple kind of meshes into the blue, the nuclear membrane or nuclear envelope becomes the membrane of the endoplasmic reticulum, and you have both rough areas of endoplasmic reticulum that have the ribosomes, and then in the back you can sort of see where there's no more ribosomes, and that would be smooth endoplasmic reticulum. So this is adjacent and connected to the outside of the nucleus via the nuclear envelope. I hope that makes sense. Okay, so let's start with what is the function of the endoplasmic reticulum. It really breaks down to the different types in terms of what their functions are. Smooth endoplasmic reticulum that lacks ribosomes is going to be synthesizing things besides proteins. Rough ER, which has ribosomes, is going to be synthesizing proteins. So as shown here, the various things that the smooth ER can synthesize include lipids. It also is involved in metabolizing carbohydrates, detoxification of various foreign substances that could be harmful to the cell, and storing calcium ions. Notice there's nothing here to be said about proteins. So what I really want you to remember is that rough ER synthesizes proteins. These proteins are specifically going to be leaving the cell. They're going to be exported from the cell. Versus the smooth ER synthesizes other things that might also leave the cell. Okay, now after the materials are synthesized by the rough or smooth ER, or these other processes occur, let's take a look at what's coming up next. There's no direct connection between the endoplasmic reticulum and the Golgi apparatus here in kind of like a teal color. So because there's no direct connection, the transport happens through vesicles. So like these little kind of cups that look like they're filled with food, those are the things that are going from the ER that have been synthesized, or it could be even waste products like through the detoxification of the drugs and toxins. Those could be the waste products from that process. So anyways, they're going from the endoplasmic reticulum to the Golgi via these transport vesicles. Now what's happening in the Golgi? Let's zoom ahead to that. Okay. Sorry, these aren't in the order that I was thinking I would talk about them. So the Golgi apparatus, it receives those packages in the form of the vesicles. So it's sort of like UPS of the cell. It's going to receive those packages. It's going to sort them. It's going to kind of put a stamp, a molecular stamp that indicates the destination for that package. Where are those materials supposed to end up? So Golgi is a big job for the cell. And when you look at a diagram of the Golgi, there's a receiving side of the Golgi, or the cis face of the Golgi, and the shipping side of the Golgi, or the trans face side of the Golgi. So the cis face will always be the one that's closer to the endoplasmic reticulum, and the trans face will be the side that's facing the cell membrane. Now, the vesicles that we're looking at right now, do not confuse those with vacuoles. Vesicles are much smaller typically, and we'll talk about different kinds of vacuoles likely in a different lesson. Okay, let's go back and see how we've come with the endomembrane system so far. So now we have this, this vesicle leaving the Golgi, and if it ends up fusing with the plasma membrane, then those materials that it contained can be released from this cell, exported from the cell. Notice that this is a very important thing to notice here. The membrane of that vesicle fuses with the plasma membrane so that it's actually recycling those lipids of its own bilayer that surrounds that cell. These are membrane-bound organelles, so that membrane gets recycled. Nothing goes to waste. And that's why one of the cell structures that is involved in the endomembrane system is the plasma membrane. So there's one we haven't seen yet, the lysosome, and this one is not always involved. It really depends if it's involved, because it's going to be the kind of like the cleanup crew. If there's, say, like a food vacuole that's engulfed by the cell, made by its own membrane here, it's like a invagination or pinching off of the own cell membrane of that cell that engulfed the particle. This could be like a single-celled eukaryote, like a paramecium, or macrophage in our body that's part of our immune system. Lysosomes will fuse with those food vacuoles, and in doing so, deposit these digestive enzymes that help to break down that food, and ultimately that waste will be exported from the cell, much like we saw with what happens to the vesicles. They fuse back with the cell membrane, releasing those materials, like here would be waste. We don't want that in the cell. and thus the cell can go on and have that membrane recycled back to its own plasma membrane. So in summary, here's what I just talked about with regard to the lysosome. We'll study this a bit more in our next chapter, actually, on the cell membrane. We'll revisit this when we talk about methods that cells can obtain and release materials through, for example, this phagocytosis, or the engulfing of that food particle. Well, that's all I wanted to say about the endomembrane system and the nucleus. So these are some pretty major organelles. Be sure that you're familiar with them and understand that these are only found in a eukaryotic cell.